So the cortical granules contain molecules, some of which are enzymes, that leave the cell after they fuse, and they change the nature of the zona pellucida.
How about the proteins
On the oocyte, those are affected primarily by the fast block. Alright, so this is happening at the same time that our egg is completing that second meiotic division and forming the oocyte. Alright, so back to this picture that we looked at earlier. So the sperm has come in, it binds to the receptor, to the molecule, to the integrin protein molecule. That triggers the second meiotic division. And is anybody in here doing the infertility case study? Okay, so with insertion of the sperm directly into the egg via the little long skinny pipettes, that itself was found, like I said, kind of by accident, to trigger this second meiotic division. All right, even though there's no receptor binding. Okay, it's the change, the opening up of the plasma membrane with that pipette will trigger that. So now the two nuclei of the sperm and the egg move together. So the second meiotic division has already occurred. Here's the second polar body. And in this diagram, the first polar body underwent division 2. Okay, it doesn't always happen, but in this case it has. So in that case, what would be the chromosomal pattern of those cells resulting from the first polar body undergoing division? N as well. Great, you guys are grabbing onto that. Okay, so now the two N chromosomal material of the egg and the sperm come together, interact. We prepare for a mitotic cell division, and we get our 2-cell, 4-cell stage. So the zygote is the term that is utilized for the egg, that is the oocyte plus the sperm that have united to form a new cell with new chromosomal material. Okay, so once those two pronuclei move together and associate, and they'll immediately prepare for that first mitotic division, and that is a zygote that is doing it. All right. So we have the 2-cell stage that's shown in this model over here. Right, 2-cell stage. Notice the zona pellucida is still around it. First polar body and second polar body. So now these 2 cells, what's their chromosomal pattern? 2N, regular diploid cell, no parentheses. Okay. The first polar body is going to be 2 parentheses N, and the second polar body is going to be N. And if you understand that, then you've got it. Okay? Now, we have a 4-cell stage. What's the chromosomal pattern of each of these 4 cells? 2N. And every body cell, placental cell, anything that amnion, anything that forms from these is going to be 2N. The only time we see N is when this person grows up and its egg or sperm start undergoing meiosis. All right, and those are the only cells that are going to get back to any form of a haploid cell. All right, so as this is moving through the oviduct, it's undergoing all of these different cell divisions, and it forms a solid ball of cells 3 days after ovulation. So what day is ovulation normally? Day 14, all right? So by day 17, beginning of day 18, depending on where in that 24 hours the egg was fertilized, we have the morula. Now, you can see that we start to have a different environment for each of these cells. They're all 2N. Some of the cells are going to become the amnion. Some of the cells are going to become the embryo. Some of the cells are going to become the placenta. Part of the functioning placenta, a part of the non-functioning placenta, and yet they all have the same 2N chromosomes. Even in the embryo, all right, some of those cells are going to become nerve cells, and some of those cells are going to become muscle cells. Some of the muscle cells are going to be smooth, some are going to be skeletal, some are going to be cardiac, and they all still have the same 2N chromosomes. Right? If we decide to clone this individual, we can take those 2 N chromosomes from a muscle cell and put it into an egg, and they will form placental tissue and all that stuff all over again. All right, so what's the difference? It's the chemical environment. And you can see that just in this morula, we have a different chemical environment. The cells on the outer surface are getting a higher concentration of the chemicals in the reproductive fluid of the oviduct and the uterus. The cells that are in the center of that morula are getting less, because it takes longer to diffuse through all those other cells. And that seems to be the key difference on what cells become what. All right? Quite a few years ago, because I read about this in the early 80s when I was in graduate school, some really fun developmental experiments were done where the chicken embryo, which takes less than a month to develop, they would peel off the egg shell and do surgery on the chicken embryo, and then cover it back up again so it wouldn't evaporate and shrivel up and die, and do microscopic surgery at a certain number of hours. They would take a section of the embryo of the chicken from an area that would normally develop into the wings, and they would cut it out and then make an opening in the back and put it right over the tissue that was forming the developing spinal cord, and then let it completely develop into a chick, and it would be a normal chicken. Wait just 2 or 3 hours and do the same surgery. Take tissue, not on the same chick, but on the same age. Take tissue from the area that would become the wing. It still didn't look like it. It just looked like the regular, looked like a human embryo for all of that. Put it in the back, let the chick develop, and a wing would grow out of its back. The cells had differentiated and shut down the genetic material that made it form any other tissue but wing tissue. Just in that short time, differentiation was occurring just in those few hours. Okay? So we start to see those changes in what cells do based on orientation, based on location and placement to other cells. So by the time our morula moves through the oviduct and moves into the uterine cavity, it's become hollow. By about this time, hatching occurs, and hatching is the breakdown of the zona pellucida and the release of the free cells of the blastula, and because it doesn't implant with the zona pellucida, all right? So the molecules of the blastula interact with the uterine cavity. Implantation starts about day 7 to 9 after fertilization. So in our uterine cycle, our menstrual cycle, that would be around day 22, day 21, 22, 23. If you look at the hormonal cycling, that's about the peak of progesterone, okay? Normally the progesterone would start to fall, we'd have separation of the stratum functionalis and stratum basalis, and any implanted tissue would be lost. So what's the hormone that maintains the corpus luteum, those follicular and theca interna cells in the ovary? What keeps them continuing to make estrogen and progesterone to maintain the thickness and blood supply of the endometrium? What hormone is produced when the sperm fertilizes the egg? What's the hormone of pregnancy that women test for?
HCG.
HCG. Anybody know what HCG stands for?
Human chorionic gonadotropin.
Okay, human chorionic gonadotropin. Chorion is fetal tissue that is part of the placenta. Okay, so that's what we're going to describe next as implantation. So HCG is human chorionic gonadotropin. So that will maintain the corpus luteum of the ovary for about 3 months, and then the placenta secretes enough estrogen and progesterone to keep the uterine tissue viable so that the placenta isn't lost. If that doesn't work, then a woman may undergo a spontaneous abortion because there's a separation of the stratum functionalis and stratum basalis. All right, so I'm going to take us through implantation, and then we'll take a break. Do you guys want to do your case study then or after the second break? And we're going to come back and just do about 35 minutes of embryo development. So think about it. Let me know. We're going to look at the same pictures. This time we're focusing on implantation. When we come back after break, we're going to focus on what happens in embryonic development. All right? So we're going to focus on this aspect here. Normally, implantation takes place on the posterior surface of the uterine wall, the body of the uterus. Okay? Up in this area. If implantation takes place closer down to the internal os, then bleeding can occur, placentia abruptio, where part of the placenta develops over the internal os, and then as the uterus gets larger and larger, it starts to separate and doesn't get its vascular supply. All right, so usually is up higher on the uterine wall. So this space over here is the uterine cavity. All right, here's the uterine surface. And remember that we have uterine glands that are lined with simple columnar epithelium. They become rich in glycogen, which is starch, and provides nutrients for the cells as they're being implanted. And this layer of the endometrium would be what layer? Stratum functionalis. Okay? The base of the glands is found in stratum basalis, but at this time our endometrium is very thick, and so most of the endometrial lining is the stratum functionalis with a very rich blood supply. This would be uterine cavity here. So over here would be the other part of the uterine cavity, also lined with stratum functionalis. And I'm going to be erasing this in a series of drawings. As the blastula or blastocyst kind of tumbles its way along the uterine wall, by about day 7 or 9, it starts to attach to these columnar cells, and it doesn't eat them, it doesn't digest them, but it breaks the cell junctions that holds them together. It kind of pushes them apart and migrates into the endometrium. And let me put the next picture up while I draw this in diagram.
And you said it was simple columnar?
Uh-huh. All right, so up here I'll draw the free blastula. These are a single layer of cells, identified at first just as trophoblast cells. What would be their chromosomal pattern? 2N. You're catching on. And on the inner aspect, inside our hollow blastula, is a cluster of cells known as the inner cell mass. And those are also 2N. Okay? The trophoblast is going to develop into the placenta, what we call chorion tissue. The inner cell mass, this is what we're going to follow now. The inner cell mass is going to form the amnion, the embryo, and yolk sac. Okay? But all of their chromosomes are the same, whether it's a trophoblast cell or an inner cell mass cell. They all have have the same chromosomes.
Professor? Sorry, what is the chorion?
Chorion is tissue that's part of the placenta.
Okay.
Okay, you'll see in just a moment. I'll be describing it. So our blastula settles onto the surface of the uterus and starts to penetrate into the stratum functionalis. Again, our cytotrophoblast cells. Now that these will continue to multiply, but will form a single layer. Those cells that are in contact with the stratum functionalis fuse. Their plasma membranes join together, and we have one large massive cytoplasm, like one big, huge amoeboid-like cell, all right, without individual differentiations. The nucleus, the cytoplasm, everything would be shared. And so our cytotropoblasts on this side start to multiply. So these are also all cytotropoblasts. And then some of them start to fuse. And they form this large single structure. Syncytial means fused, okay, with. So cells that are together. And so this is identified as syncytial trophoblast. And it forms these like kind of flowing fingers and kind of penetrates its way through the stratum functionalis. As it does so, the whole entire blastula sinks deeper and deeper into the stratum functionalis. As this is occurring, as this is sinking in deeper and the syncytial trophoblast is spreading out and extending its finger-like structures, the connective tissue of the stratum functionalis undergoes an inflammatory reaction. The fibroblasts, making the collagen fibers, enlarge and get round, and that's known as the decidual reaction. That occurs around the entire uterine cavity. So all of the stratum functionalis of the entire uterine cavity undergoes this, just deep to the epithelium, undergoes this inflammatory reaction. And so all of the maternal connective tissue of the endometrium now is called the decidua. Okay? Not the stratum facialis, but the stratum functionalis part of it is all called decidua. So I want you to recognize decidua as being maternal tissue and chorion, we'll get to in a moment, as being fetal tissue. So any questions on this slide before I move to the next step, which shows our blastula sinking even deeper. I'm not going to talk about inner cell mass right now. We're just ignoring that for now. Okay? Okay, now it's sunk in even deeper. So rather than moving this direction, I'm going to redraw it by bringing my endometrial tissue over this direction. Okay? So now this is going to be the simple columnar epithelium of my uterine surface. Here is the uterine cavity. Now that this is completely enveloped and it's kind of, the uterine cavity is closed in over it, the cytotrophoblast around the entire, let me just put my inner cell mass cells in here to give us some reference. The cytotrophoblast around the entire blastula are multiplying, and the syncytium now is also found completely surrounding the enveloped blastula. So here's the syncytium, here's the cytotrophoblast. Okay? As the syncytial expands, it comes across the maternal blood vessels, the spiral arteries that have proliferated within the stratum functionalis. And it actually invades those, and it opens them up and starts oozing up the sides. Like you imagine a cement duct or a cement tunnel and slime growing along the sides. So now the syncytial trophoblast is directly exposed to maternal blood. Okay? And these lacunae are those pools of maternal blood. Now, two things occur in the development of the tissue. At the end, towards the deepest part of the syncytial trophoblast, we start to see more organized divisions. So more distinctive branching of the syncytial trophoblast. The pools of blood are now exposed in contact, directly in contact with the syncytial trophoblast. So this is maternal blood coming from those spiral arteries, kind of forming a pond or a lake, and these syncytial trophoblast branches are going out into that. And that's going to be mainly primarily down around this area, because here there's not many blood vessels between the blastula and its surface. In addition now, the cytotrophoblasts are going to start to form columns of cells that are going to migrate down into these fingers of syncytial trophoblast. These fingers are the villi. Okay? The cytotrophoblasts undergo changes and form fetal connective tissue. Even though our embryo is here, this is extra-embryonic. It's connective tissue outside the fetus, but it's still fetal tissue, right? It's still the new DNA. So these cells are going to become fibroblasts and collagen fibers. And so we're going to see a finer branching like the little leaf rootlets. If you were to pull a root out of a little plant out of the ground and wash it and look at it under the microscope, you see these really fine rootlets and branches. Okay, I'll leave this down here for the old portion of it. And then these finer branches are going to have the connective tissue that develops from the cytotrophoblasts. Fetal fibroblast, fetal capillaries, blood vessels also will form and come up to the, at this time we're getting blood supply from the fetus. So if we look at a cross section of one of these branches, we're now moving into 3 months, 3 and 4 months or so, all right? And this core, what I've drawn as purple is shown here is this brown. This would be the cytotrophoblast that's moving down there. So this is a branch, maybe about 4 to 5 months of the placenta. One of these branches right here. If I were to cut that in cross section, so make a cut through there, that would be my syncytial trophoblast. One big oozing plasmodium, a bunch of cytotrophoblasts, and the number of cytotrophoblasts decreases with each set of branching. So in the term placenta, you're just going to have 3 or 4 scattered along, not a whole lot. Then we would have our fetal fibroblasts and collagen fibers and blood vessels. And this entire thing would be in contact with maternal blood. It would be like taking your finger, so the skin on my finger would be the syncytial trophoblast, and then inside that would be blood vessels, and dipping it down into a cup of blood. That's the maternal blood, and the oxygen from the maternal blood would move into the fetal blood, be carried up the vessels and into the umbilical cord to the baby. Okay? And that exchange of nutrients would be occurring across that surface. Now you can see in this first picture that those branches of chorion, so chorion is this fetal connective tissue, all right, surrounded by the syncytial trophoblast. These branches of chorion at first develop all the way around our embryonic tissue. But because they're so close to the uterine surface here, they can't develop very well. So they might find 2 or 3 branches where these form 15 or 20 smaller and smaller branches, all right? So the entire fetal component here is chorion. On this side where we have all of the branches, it's known as chorion villi. So on the deep side, the placenta side. This area here, close to the surface, not very many branches form. And this is called the smooth chorion. So this is known as chorion levi. You might see it spelled L-A-E-V-E. There's different variations. All right? That means smooth. And smooth has an S. So the next slide will show that differentiation. So here's the uterine cavity, kind of flipped from the direction I drew it. Here's the uterine cavity. This is going deeper and deeper towards the myometrium, but still in the stratum functionalis. Here is the chorion. The blue here is showing where the maternal blood would be. And here is the chorion levi. See how it's not very branched? Here is the chorion villi, which is highly branched. Increased surface, more blood supply. That's what the developing embryo and fetus needs. Okay, so then we get to this appearance, and you can see much more clearly chorion levi versus chorion villi. Right? Same thing with this model. So in this model right here, the blastocyst has yet to be completely covered over by the uterine epithelium. It hasn't completely gone underground. So this would be the uterine cavity. All right, it's implanting in this direction, similar to the way I have it in my earlier picture. These cells would be cytotrophoblast. Going around here would be syncytial trophoblast. All right, and then the inner cell mass. And this would be the decidua, or syncytial trophoblast of the mother, all of this tissue that I showed you right here. Now, it descends, is completely enveloped. So in this image, in the same orientation, it's completely covered by uterine epithelium, but it's now starting to bulge up into the uterine cavity. Still covered by the surface of the uterus, all right, but now bulging, as you can see in this diagram, into the uterine cavity. So it would match, this point like this. So this would be uterine cavity out here. This is deeper into the stratum functionalis towards the myometrium. Okay. Notice that the dark reddish brown, highly branched here, not highly branched here. So chorion villi, this is what's actually forming the true placenta. Chorion levi, the reddish brown that doesn't have very many branches. There's not much blood supply here, so there's no point in branching. All right, so that's fetal tissue. Chorion villi and chorion levi is fetal tissue. Now, remember I talked about the decidual reaction? So that's the stratum functionalis, the part that gets thicker and sloughs off. It gets thicker and sloughs off as we do the uterine cycle. We have 3 areas of decidua. Okay? Stratum functionalis has undergone those changes. One area is associated with the chorion villi. It's a little confusing because we use that term basalis again and we're talking about stratum functionalis. But that area of the stratum functionalis associated with the chorion villi is known as decidua basalis. Even though the S and the decidua should be there, even though it's part of the stratum functionalis. Because remember, basalis doesn't undergo changes. So that part of the stratum functionalis associated and contributing to the maternal blood with the chorion villi is decidua basalis. Now part of that stratum functionalis is captured up here as the embryo is growing and bulges upward. So here is the rest of that roof of stratum functionalis. And so associated with the chorion levi, we have decidua capsularis. All right? So chorion villi is with decidua basalis. Chorion levi is with decidua capsularis. Now all the rest of the uterine cavity has stratum functionalis as well. No direct association with providing nourishment to the developing embryo, and it's called decidua parietalis, or the parietal aspect. Right? All stratum functionalis undergoing the decidual reaction, but 3 different locations. So decidua basalis, decidua capsularis, and then decidua parietalis. Right? As the developing fetus grows, the uterine cavity is almost obliterated as the 2 uterine epithelial tissue come in contact and actually touch each other. So I always had the idea that the fetus was actually in the amniotic sac, and the amniotic sac grew and developed and touched the other side. But it's really deep to the uterine surface, and the 2 surfaces come together. All right? So here is just about term. I don't know what happened to the baby because obviously there was no birth here. Now it's still closed. But anyway, baby's gone. The amniotic membrane is developing from our inner cell mat, so we'll talk about that after break, all right? But here is the chorion levi. Attached to it is the decidua capsularis. Here is the chorion villi. Here is the decidua basalis, all right? And all of this is decidua parietalis. Anyone heard of chorionic villi sampling? All right, now I was 38 when I got pregnant with my twins. Since I was over 35, I had an amniocentesis. Because they were fraternal twins, I actually had to have 2. Sampling of getting amniotic fluid from each of the amniotic sacs. In fact, that was when we found out we were having twins. They did ultrasound to find out where the baby was to put the needle, and the technician said, "Congratulations." And we were like, "Why?" And she said, "Well, you're having twins." And I had 4 ultrasounds by then. I'd actually asked my OBGYN the first time I went in there, "Please tell me I'm not having twins." And he said, "Well, why do you think you are?" I said, "Well, the home pregnancy test was just much more stronger than I expected it to be." So he listened to the fetal heart rate, did a vaginal ultrasound, and said, "No, I only see 1 baby." That was at 8 weeks. At 9 weeks and at 12 weeks again, I had ultrasounds and just 1 baby. 14 weeks, I was going in for the amnio, and so he didn't do one. And then at the ultrasound, unfortunately my husband was with me because that was quite a shock after being told over and over and over that we weren't having twins. And so the doctor said, well, I'm going to be calling your OBGYN. I said, I'm doing it from the card. And so instead of calling, we actually went over to his office and we had the photograph from the ultrasound. And you can see the two heads, head A and head B, they were marked. And there weren't any patients in the waiting room, so he came out. And we just come back from a 3-week trip to Asia. And so he said, so, you know, how's the ultrasound? And I didn't say a word, I just gave him the picture. And he swore and threw it down on the table and said, I'm sorry. And I said, well, if you had known that we were pregnant with twins, you wouldn't have allowed me to go on the trip. And I went back to where I was born in Burma, and my dad was born in China. We took him with us, and so it was a wonderful trip that I wouldn't have missed for anything. But because they were fraternal twins, I had 2 amniotic sacs. Sometimes testing is needed to be done earlier, all right? At 7 or 8 weeks, if we go back to this slide, amniotic sac, way up there. Most likely you would kill the embryo trying to get amniotic fluid sampling. What you do with that is you're taking cells that are sloughed off of the baby's surface and skin and looking at their chromosomal patterns. Can't do that. Pretty easy, however, to come in here and take some of this tissue. Is it going to have the same chromosomes as the fetus or embryo? Yeah. So that's called chorionic villi sampling, and it can be done much earlier and more safely than the amniocentesis. Okay. So, when a woman's bladder ruptures, it normally happens on its own. The amniotic fluid is lost, down her leg or whatever, and however, sometimes a woman's labor is induced by rupturing the membrane, and as that fluid is lost, it can trigger contractions. So when my twins were born, I was actually induced